Aloha BIOCHEM. In this second lecture on Chapter 6, we discuss the reaction rate. So in this video, we'll cover the second important aspect of chemical reactions, the reaction rate. And this concerns section 6.4 in the text. The reaction rate is defined as the rate of product formation. So in a chemical reaction, you have reactants disappearing and products appearing, and the rate is the rate at which the products appear. Let's see what this means in an example. Here we have ethylene reacting with water to form ethanol. And from the coefficients, 1 and 1, you can see that one molecule of ethylene combines with a molecule of water to form an ethanol molecule. So this is a combination reaction. Now, let's imagine the reaction taking place in a vessel. And here I've drawn 5 molecules of ethylene and 5 water molecules. And if we watch this reaction occur over the course of a couple of seconds and we observe the product appearing, we can talk about the rate. So in the first second, you can see some of your reactant has disappeared and some of the product has appeared. And so maybe these two molecules combine to form a product, and maybe those two combine to form another product. So in the first second, we have formed 2 product molecules. Now in the next second, we gain another product. So maybe those two combine to form another product molecule. And now that we've observed the products appearing, we can see what the rate is. The rate is usually the amount of product that appears per second. Okay, that's how it's discussed. And so in the first second, we have formed 2 product molecules, and so the rate over the first second is 2 molecules per second. And in the next second interval, we've gained another product molecule, but we've only gained 1 here. And so the rate in this interval is 1 molecule per second. Now, it looks like the reaction went faster in the first second than it did in the second second. And that usually happens. When reactions occur, they slow down over time. And the reason's because the rate depends on the amount of reactant that you have in the container. And as the reaction progresses, you get less and less reactant, and so the reaction slows down. And that's actually the first factor that we'll discuss that affects the rate. The rate depends on the concentration. When you have more molecules of reactant per unit volume, it's faster. So imagine a very dilute solution versus a very concentrated solution. When you have more reactant molecules in the container, it goes much more quickly. And the reason is because the molecules are colliding together more frequently. If you go back to our little combination reaction, you can only have this reaction occurring every time an ethanol molecule collides with a water molecule. Okay, if they're not colliding together, they can't combine to form a product. So the reaction depends on the collisions that are occurring. And when there are more reactant molecules in there, you're going to get collisions occurring more frequently, and so it'll be faster. Now, as an aside, since we're talking about concentration, let's just make a note that concentration is usually expressed in units of molarity. Okay, we don't need to talk about such units for the rest of this lecture, but you'll need it in subsequent lectures, so let's just mention it now. Molarity units are defined as the moles of solute divided by the liters of solution. And so the units are moles per liter, or capital M for short. You'll see other units out there like percent, and there's different types of percentages like percent mass over volume, percent mass over mass, percent volume over volume. So if you're working in a medical lab and someone hands you a 3% NaCl solution, that means 3 grams of sodium chloride per 100 milliliters of water. So that's a 3% M over V saline solution. So just be aware that there are other units out there, but in the chemical lab we use molarity primarily. And as a quick example, a 2.5 molarity NaCl solution has 2.5 moles of NaCl dissolved per liter of solution. So if you have 1 liter of solution, there would be 2.5 moles of NaCl dissolved. And you can see if you take the 2.5 moles divided by the 1 liter, you get 2.5 M. And if you have 2 liters of solution with 5 moles of NaCl dissolved, then you can see 5 over 2 gives you 2.5 M again. So this has the same concentration as that. And you can see, this liter and the liter down there have the same amount of sodium chloride. So it's the same concentration. Okay, so be aware that concentration is expressed in units, and there are different units that you'll see. The next factor that affects the reaction rate is the temperature. And here, hotter temperatures cause the reactions to occur more quickly. The reason for that is at hotter temperatures you have faster molecular motion. Imagine molecules flying around inside of a cold container at 100 Kelvin versus a warm container at 300 Kelvin. Well, in the warm container, everything moves more quickly on the molecular scale. Molecules are flying around and they're also vibrating more quickly. And when you have faster molecular motions, you also have collisions occurring more frequently. Okay, they're colliding with the walls, the molecules, and they're colliding with each other. So hotter temperature means more frequent molecular collisions, and the reaction would go more quickly. As a second example, let's look at the following reaction. This is an isomerization reaction. So here a single molecule just rearranges, and so the product and reactant molecules are isomers of one another. This is the isomerization of methyl isocyanide. So you have a methyl isocyanide over here and the methyl cyanide. So you can see in this reaction, these two atoms just kind of rotate around. So the nitrogen-carbon bond breaks and you form a new carbon-carbon bond. Okay, now let's imagine this transformation taking place on an energy diagram. You remember energy diagrams plot the energy of the molecules as they change from reactants into products. Okay, so as the reaction progresses, how does the energy of the molecules change? Okay, now your reactant molecule has a certain energy, okay, and your product molecule has a certain energy as well. And it looks like in this case the product is lower in energy than reactant. So this is apparently an exothermic reaction. So if you start with more energy and you end up with less energy, then that excess energy is released as heat. Okay, so this is an exothermic reaction, but that's beside the point. Let's imagine the reaction, how it proceeds. We know the carbon-nitrogen bond needs to break, and that's going to cost energy. Okay, when a chemical reaction occurs, bonds break, which costs energy. And then we know that the new carbon-carbon bond forms, and you get that energy back. And so that's where you get the energy back. Now, the transition state for this reaction is kind of like the halfway point. Okay, it's kind of like where you got, the bonds have been broken to a certain extent, so the carbon-nitrogen has been broken. Then you have this new bond forming to a certain extent. So it might look like that. It probably does. And to get from the reactant to the transition state, this is like the tipping point, requires the activation energy. Okay? Now, in order for the reactant to convert into this, tipping point structure, this transition state structure, what probably happens is the bond over here kind of flaps around. Okay, molecules undergo vibrations, all right? And this molecule is constantly vibrating. Now, if the temperature is higher, not only do molecules move faster, but they vibrate more vigorously, stronger molecular vibrations. So this molecule is vibrating more vigorously. It's getting to this transition state structure, more frequently. At colder temperatures, the bond might not vibrate so much, and so it's not going to reach this transition point and turn into product as often. So colder temperatures, the molecule doesn't vibrate as much. It's not getting over the barrier as frequently, but higher temperatures, it's vibrating, more quickly and more vigorously, and so it gets over the barrier. Okay, so the transition state is reached more frequently. So the reactant, if it's under hotter temperature, it approaches the transition state easier. It reaches the transition state and it forms the product. All right, so temperature, hotter temperatures means faster. The third aspect, or the third factor which affects a reaction, is a catalyst. And catalysts are kind of interesting. What they do is they provide an ideal surface on which the reaction proceeds. And I'll show you two examples of reactions that occur with the help of a catalyst. In the first one, we have ethylene reacting with hydrogen to form ethane. And you can see that you have hydrogen atoms being added to each carbon to form the product. This reaction is slow without the help of a catalyst, but in the presence of palladium metal, it occurs very quickly. What the metal does is it provides a surface on which the other two molecules attach to. And once they attach to the surface, the reaction occurs very quickly. So here's a picture of kind of how it happens. So you have the molecules flying around in space, and they would react very slowly up there, but if you put a palladium metal in their presence, then the molecules become attracted to the metal. And so the hydrogen molecules get attached to the metal, and the metal, helps split apart these hydrogen molecules into hydrogen atoms. Okay, and so you have some hydrogen atoms attached. Now the ethylene molecule also attaches, and if you have both attached to the surface, then it becomes easy for these atoms to add to the ethylene molecule. And so, you have one has added to this carbon, and that detaches this carbon from the surface. Then you have another adding to that one, and then you get the product molecule. Okay, and that's how it happens. You have the reactant molecules become attached to the same surface. The reaction happens at the surface, and then they detach from the surface. And catalysts are very useful because they're not used up in the process. You still have the palladium left over at the end, so you don't run out of catalyst. The energy diagram of a chemical reaction helps show how the catalyst speeds it up. In this diagram, you can see the red catalyzed curve has a lower activation barrier than the blue uncatalyzed curve. You remember, as reactants convert into products, they need to proceed through the transition state. And the transition structure has partially broken and partially formed bonds. It's an unstable structure, and it's hard for the reactants to achieve that structure. It takes quite a lot of energy. But with a catalyst, what that does is, by providing a surface on which the reactant molecules attach, once they attach, the structure of those molecules are deformed in such a way as to sort of resemble what the transition structure might look like. So the enzyme helps kind of provide some of the the energy needed, and so it's a lower energy that the reactants need in order to achieve that transition structure. So a catalyst lowers the activation barrier, and that makes for a faster reaction. Uncatalyzed reactions have a higher barrier, and they yield slower reactions. Another example is the decomposition of lactose. Lactose is a sugar, and it can break down into galactose and glucose. And the way this reaction happens is a water molecule reacts right here where this oxygen is. And so the lactose molecule breaks apart into two smaller molecules. One is galactose and the other one is glucose. These are very similar simple sugars. They have the same chemical formula, in fact, but the structure is slightly different. Now, this reaction occurs inside of the body, but it's very slow without the help of the proper catalysts. And catalysts in the body are called enzymes. And the enzyme which helps this reaction occur is called lactase. Now, this works the same way that other catalysts do, is it provides a surface on which the reaction proceeds. And here you can think of the reaction occurring as follows. So you have your enzyme, which is a very large, molecule, and being very large compared to the reactant molecule, it provides like a surface. And a certain portion of this surface seems particularly attractive to the lactose reactant. Okay, so the lactose reactant attaches to a certain portion of the lactase enzyme. This is called the active site where it gets attached. And once it's attached, the enzyme molecule, kind of helps break the molecule apart. The enzyme has certain vibrational motions that it undergoes, and these vibrational motions actually help the reaction proceed. And so as soon as it helps break the reactant molecule apart, and also there's a water molecule involved, remember, and so the water molecule has to react as well, and the enzyme can help that happen as well. And then you get the two product molecules, and then they detach from the enzyme, and you get the enzyme back. Here's another picture of how it might happen. So at the beginning, you have your enzyme molecule and your lactose molecule. It attaches to the enzyme in the active site of the enzyme, and then through the vibrational motions of the enzyme, the reaction can occur. So it involves a water molecule too, and then the molecule breaks apart into the two product molecules, and they detach, and you get your enzyme back. So enzymes are very rich in structure. Here's a nice picture of that lactose enzyme. You can see it's a quite big molecule. It's huge compared to the reactant molecules. Okay. Enzymes have an interesting structure. They're actually made of amino acids, and it's hard to see here, but this is actually a long chain of amino acid molecules that are attached together. So you can imagine a bunch of amino acids attached together, in a long chain. Okay, and there's different types of amino acids, and so they attach together in a long chain, and this chain sort of, folds up into the nice, interesting structure. Sometimes you get these helical structures, sometimes they zigzag back and forth, and so you end up with drawings like this, which represent enzymes. And so you see a lot of these nice helical structures and the zigzags back and forth. And this is the same lactase enzyme molecule, okay, that we just saw up here. So there you go with all the atoms, but it's kind of hard to see everything, and so maybe drawings like this can help you see the structure a little bit better. But anyway, this enzyme molecule provides a surface on which the reaction proceeds. All right, this reaction is discussed in your textbook, by the way, as well. So here's figure 6.3 in your textbook that discusses lactase. So here's your lactose sugar that you'll find in milk, and once you consume this, your body likes to break down sugars, and the enzyme which helps it do it is lactase, and the molecule attaches to the lactase enzyme, and then it breaks apart into the two simple sugars, glucose and galactose, and then it detaches and you get your enzyme back. So enzymes are pretty cool. And that's it for this lecture. So stay tuned for the next one on equilibrium. Aloha.